Welcome to Biology, the scientific study of life. Biodiversity and human welfare. Why should we care about the loss of biodiversity? Perhaps the purest reason is what E.O. Wilson calls biophilia. Biophilia, our sense of connection to nature and other forms of life. The belief that other species are entitled to life is a pervasive theme of many regions and the basis of a moral argument that we should protect biodiversity. There is also a concern for future human generations. Is it fair to deprive them of Earth's species richness? Paraphrasing an old proverb, G.H. Brundtland, a former Prime Minister of Norway, put it this way: we must consider our plan to be on loan from our children rather than being a gift from our ancestors. But species diversity brings us many practical benefits as well. Benefits of species and genetic diversity. Many species that are threatened could potentially provide crops, fibers, and medicines for human use, making biodiversity a crucial nature resource. In the United States, 25% of all prescriptions dispensed from pharmacies contain substances originally derived from plants. For example, in the 1970s, researchers discovered that the rosy brinkel, which grows on Madagascar as inland of the coast of Africa, contains alkaloids. Notice this is the rosy periwinkle, a plant that saves lives, that inhibit cancer cell growth. The result of this discovery of most people with either of two potentially died from a form of cancers, Hodgkin's disease and a childhood leukemia is remission. Madagascar is also home to 5 other species of periwinkles, one of which is approaching extinction. The loss of these species would mean the loss of any possible medicinal benefits they might offer. The loss of species also means the loss of genes. Each species has certain unique genes, and biodiversity represents the sum of all the genomes of all organisms on Earth. Such enormous genetic diversity has the potential for great human benefits. Consider the historical example of the polymerase chain reaction, PCR. The gene cloning technology based on an enzyme extracted from thermophilic prokaryotes from hot springs. Currently, corporations anticipate using DNA extracted from the prokaryotes in the numerous hot springs in Yellowstone